Our project deals with extracellular specific immunity. Extracellular because it's not cell-mediated, meaning it takes place throughout the body as opposed to in any one particular cell, generally in the blood. For this, it's often called humoral immunity. Specific, as opposed to non-specific immunity, because it targets certain antigens and viruses rather than a more generalized immune response. Before we begin, we'd like to give you a brief overview of the types of cells involved in this particular response. Chief among them, the B cell. B cells are charged with creating specific antibodies to recognize specific antigens and elicit a specific immune response. Crucial in this reaction is the presence of the MHC2 molecule on the membrane of the B cell. Once a B cell is paired with its cognate antigen, it's considered a plasma cell. Plasma cells will continue to secrete antibodies as part of the immune response. Most plasma cells will die after 2 to 3 days. The 10% or so that continue to live on afterwards survive in the form of memory B cells. Memory B cells are crucial to immunity in that they will remember that particular virus and retain all the tools necessary to conquer it should it ever attempt to reinfect the host. Helper T. Helper T cells are considered immune response mediators, meaning they're basically the bosses of the entire immune operation. They cannot kill pathogens or infected cells themselves, but they can direct other cells to do so. How it works is when a helper T cell is contacted by an APC, antigen-presenting cell, it will release cytokines, which are small chemical protein messengers similar to hormones, to elicit one of two responses. Either it will elicit a cell-mediated response, as you see on the right of this diagram, or it will initiate a humoral response, as you see on the left side of this diagram, mainly involving B cells and plasma cells. This will be the focus of our presentation. HIV, interestingly enough, attacks the helper T cells themselves, which neutralizes the entire immune response by destroying the heads of the operation.
Antigen-presenting cells, or APCs, are messengers for helper T cells. Macrophages are one type of APCs. When they recognize a pathogen or its antigens, they completely ingest the pathogen through phagocytosis. Class II MHC cells are on the surface of all APCs. From there, the macrophages present helper T cells with the antigens so that the helper T cells can initiate a humoral immune response with cytokines. Another type of APC is a dendritic cell. It can be found in places where infection is most likely, like mucous membranes, the dermis of the skin, if you remember the Langerhans cells, lymph nodes, gastrointestinal, and reproductive tracts. When an APC processes an antigen, they migrate through the lymphatic networks from the tissues to the lymph nodes. Each B cell recognizes only one type of antigen. In a process called clonal selection, when the body detects a threatening pathogen, they make more of the necessary B cells to combat the infection. Each B cell has surface receptive sites that recognize antigens from bacteria or viruses. In clonal selection, when a B cell comes into contact with its specific antigen, it is activated. After this, it is stimulated by cytokines released from helper T cells and splits and is cloned to make more B cells that recognize the same antigen in order to fight the pathogen. The activation paired with the cytokines to stimulate the cloning is called co-stimulation. Some of the cloned cells become plasma cells and secrete a form of the antibody. Other cells become more efficient at reacting to the antigen, live for many years, and build up a lasting immunity against a pathogen. This is the idea behind vaccinations, to build up memory cells which can recognize a certain virus or bacteria so they are ready to fight immediately if they come into contact with the infection.
Once a B cell has been activated, it constantly produces antibodies. B cells could be called antibody factories of the body. B cells can only produce one type of antibody, which is meant to target only one type of antigen. The antibodies are actively produced and released throughout the body. Once an antibody recognizes its appropriate antigen, it uses its Y-shaped form to form a bond. Each antibody has a parotype that acts as a lock structure that matches only one specifically intended antigen's epitope, which resembles a key structure. This lock and key are designed to match perfectly and will not bond with any other type. Upon bonding, the antibody neutralizes and immobilizes the antigen. This prevents the bacteria or virus from attaching to body cells, and it limits the spread to nearby tissues. Once bonded, the antibody and antigen are considered an antigen-antibody complex. This complex activates the complement system, which you will learn about from a different group. The last main function of the antibody is that it enhances phagocytosis. Once the antibody has formed a bond with the antigen, the stem region of the antibody serves as a flag that will attract phagocytes. There are many different types of antibodies, but I'm only going to discuss the 5 main ones with you. You would be wise to memorize these. First off, the IgG looks like this. It is the most common of the antibodies, and it makes up about 80% of the antibodies in blood. It is the only antibody that crosses the placenta from mother to fetus, and therefore serves as the main immune protection for newborns. Next, the IgA looks like this. This one is found mainly in sweat, tears, saliva, mucus, and breast milk. It makes up about 10 to 15% of the antibodies in blood. The levels of this decrease in times of stress, which lowers the resistance to infection. IgA has a localized protection on mucous membranes. Next up, IgM, which looks like this. This one makes up 5 to 10% of the antibodies in the blood. It is also the first antibody class to be secreted by plasma after initial exposure to an antigen. The IgD is mainly found on B cell surfaces as antigen receptors, looking like this. It is very involved of the activation of B cells. It only makes up about 0.2% of the antibodies in blood. Finally, there's the IgE, which makes up less than 0.1% of the antibodies in blood. It looks like this. It can be found on mast cells and basophils. It is greatly involved in allergic and hypersensitivity reactions, but it does provide protection against parasitic worms.